Hello, Dan Guerra here from VerEvMed. Today we're going to start a series, an arc of lectures on aging. I previously published a YouTube, rather lengthy one on, I guess, sort of a propedeutic to these lectures. That is an examination of how we go about generating hypotheses to study any kind of scientific discipline or principle. And then we used aging as an example of it by looking at something called the square of opposition. So I recommend that one to you, and I can talk more about it later. But right now, let's get started. So we're VerEvMed. And what do? We're scientists verifying published evidence. And this is sort of a longer version of what our normal logo says, but let me fill it in so that you have an idea of what we cover. And it's not exclusive only to these disciplines, but we verify published evidence in the scientific literature in biochemical, physiological, and pharmacological, as well as the medical sciences. So that's our job, and that's what we do. And as clients, we would interact with you one-on-one. So, as I said, today we're going to start an arc of lectures on aging, something that is dear to everyone's heart. We all are aging. The moment we're born, we start aging, and the rate of aging is not linear throughout time. Some of us age more rapidly in a year than we might in the next 3 or in the previous 5, and others of us live to be a very long time, so our lifespan is well into the 90s, and whereas others succumb to various kinds of illnesses, maybe associated with the aging process itself much earlier than that. So we're going to start in on this arc, and let's get going. So let's introduce this. So physiological aging doesn't really proceed according to a specific genetic program. There is a genetic by environment component, and I call that a synergy. That is the two working together, right? So there's a synthesis of the association of what's in your genes and what's out there in the world, you encountering it. And there are also something called epigenomic modifications. And I'm going to give a lecture or two on epigenomics and epigenetics in some other subsequent VerEvMed. But right now, all I mean by that is that something that is supergenomic, something that is imposed upon, mapped upon your normal genomic ability to express genes. Okay? And that's because of environmental stimulation and alteration of the structure of the chromatin. Now, the rate and extent of aging, as I've alluded to, in each individual will change with age. So there is a chronological aspect to aging. And there's also what I call an asynchronous periodicity, which I just alluded to also a few moments ago. That is, in 1 year, your body or the organs in your body or the cells in a given organ or tissue can age much more rapidly than any other given year. So it's not a uniform linear process by any means. And we know this from numerous animal studies and also human studies. So to complicate that even more, and these are complications because aging, as it turns out, is tractable, but it's very complex. So to complicate these matters, the processes that drive aging are often inferred from measurements of organismal lifespan, or what we call longevity, which, although clearly related and determined in part by the nearly ubiquitous process of aging, they may be dissociated from the determinants of aging. That is, the process itself can be examined under, say, a close scrutiny microscope, and we can see how that process functions. We can see how cells age. We can see how many times they have in them to divide. We can look at repair mechanisms in the cells so they don't allow mutations, those sorts of things. Those are processes. But the determinants of aging, that is what really is involved in determining you as an individual and how rapidly you age, is not something that just is involved in gene expression or even epigenomic alteration of gene expression. So nevertheless, with all these caveats, within the framework, several general findings and principles currently drive the field that is known as biogerontology. Okay, that's the field of studying aging, right? So rather than use the word aging, scientists like to use the word senescence. Senescence is probably a better word. It's more of a principled scientific term than aging. And that's because it extracts out, or it brackets, I should say, out the idea of chronological aging, right? So where does senescence come from? It's Latin from senex, which means old man. And there's Kronos, the Greek mythological god, over there to your right. So what do I mean by senescence? It's the process of deterioration that is correlated with aging. Let's throw out a couple more definitions. Maximum lifespan. We all have been hearing about this all of our lives, right? So that it's increasing as we've been aging, actually. So the maximum number of years that a member of a species has been known to survive. That's the maximum lifespan. So the range is really tremendous if you look at all biological systems. Drosophila, the fruit fly, a model organism studying genetics, which we're going to be talking about today, genetics, not Drosophila. Humans, maybe they're 3 months. Humans, maximum somewhere around 120 years. Turtles and lake trout, more than that, maybe up to 150 years. And some trees, as we'll see in a moment, like the bristlecone pine, 1,000 or more years they can age. And so their longevity is quite substantial. There's also a term we use called survivorship, and that's the percentage of survivors in a population versus their age. So at any given age, who survives to that age, right? So again, the variation, as you might guess, because one of the variables is age, right, and the other is survival, so that's going to vary. So only a small percentage, for example, of wild animals, elk, deer, bear, for example, can actually enjoy what might be their biological senescent pattern or maximal lifespan. And that's because they die of the vagaries of environment, right? Food sources run out, water sources run out. I'm not even talking about hunting. I'm just talking about natural occurrences out there in the forest that can take out animals, or in the desert, or in the ocean, right? So you can talk about a survivorship, but that's not the same as the maximum lifespan, right? So we have to consider that too, because humans are animals, and we're living in environments that can sometimes alter our survivorship, right? So we know this because in sub-Saharan Africa, for example, or in parts of Southeast Asia, or even indeed in parts of South and Central America, there are geographical locations where the lifespan is much shorter than it is, let's say, in Europe or in the United States or Canada or even in Mexico. And that's because of this survivorship principle, right? There are other things that take out people, particularly when they're very young. Parasitic diseases being a major cause of that. Hemorrhagic diseases, diuretic diseases, things like this. And also when people get a little bit older, sometimes that advanced age will increase their chances of dying earlier because of, let's just say, falling or picking up an infection and not having the appropriate medical placement there to be able to take care of them. So that's what we talk about when we say survivorship. Now, there is a person that is touted to be the oldest ever lived. She's at least documented. Her name is Jeanne Calment. You can see that she died in 1997 there. She was 122 and a half years, roughly. And her lifestyle, that would be a big question to ask right away. Someone lived that long. First of all, what was the quality of her life? Well, she didn't die with Alzheimer's disease or Parkinson's disease or even prefrontal dementia. So she didn't have any kinds of vastly deteriorating principles and concepts of consciousness. She seemed to have most of her faculties of reasoning all the way to the day she died. That's one thing that's interesting. Now, whether or not that's correlated or even maybe a dreadful term in science, causal, would definitely be something that would be difficult to study, but because this is just one person. And even if we study all people that live to be over 100, we can't always say that dementia isn't a factor. But anyway, she was not. And also, she was not a person that ever became obese. So throughout her life, she did not suffer from many periods of time where she was overweight. Again, there's a lot of argument out there about whether or not aging is increased by an increase in body mass index. And in the later lectures, we'll tackle some of that literature, the scientific literature, see whether or not it actually substantiates that or refutes it or has really nothing to say. So what's this here? This is the bristlecone pine. It's probably about 5,000 years old. That's from tree ring analysis. It's pretty accurate, actually. And of course, this tree is totally unaware, right, So it has no consciousness that it's that old. Maybe consciousness itself has something to do with longevity. See, that's one parameter we could certainly unveil in our discussions. At any rate, 5,000 years old. It is in the White Mountains of California. It's probably the oldest living organism that we're aware of. That doesn't mean it's the only one that gets to be that old. And where does it live? It lives at about 11,000 feet, right? That's like over 3,000 meters. That's really high up in the White Mountains in California. And when it was first setting down roots, when that was a little sapling, great pyramids were being made, probably already finished being made. And because I'm sort of a student of philosophy as well as of science, I like to think about my favorite Greek philosophers. And again, this was about 2,500 years before Plato was even born. And Plato has been with us now in the literature for 2,500 years. So it's just amazing to me. Bristlecone pines themselves owe their longevity maybe to the brutal environment that they can survive in. It's not necessarily causal, again, maybe correlative. Where this particular ancient tree lives, there's a lot of desiccating winds. There's a very short growing season. There's a lot of freeze thaw every night in the soil. That can serve to diminish parasites and pests that might invade or otherwise cause that organism, that one particular tree, to die and not reach its maximal lifespan. Right? So whatever the conditions were, this tree seems to be reaching that maximum lifespan. You see, it's not a very good-looking tree. You can see that it's kind of gnarly and there's a lot of dead branches. But it's still living and it's still photosynthesizing, and it seems to be going strong at 5,000 years. Of course, it could be struck by lightning today, and that'd be the end of that. Right? So let's go back to people. There's a group of people that live on Sardinia, which is an island about 150 kilometers due west of Rome on the Italian peninsula. We call these people the Sardiniari, right? And interesting from our perspective today, men on that island tend to be some of the oldest living men that have been discovered living that long on the planet. Many people live well over 100 years there. That was a publication way back in 2001 in Science. So let's jump forward now. Here's a paper published just last month in Molecular Biological Evolution, and that's open access, by the way, if you want to take a look at this paper. And I'm just doing a real brief summary of it. But what it says is that the Sardiniari reported to possess the highest preserved Neolithic farmer genome. So we actually have the methods, and I'm going to talk about them very briefly in a moment here in a subsequent slide, to determine where your genome was inherited from, like what was the descent from a common ancestor. Okay, and so it looks like the Sardiniari definitely have a preserved Neolithic, that's New Stone Age genome. In fact, we all have that New Stone Age genome as well, particularly Europeans. The one aspect to consider about Sardinia, even though it's not that far from mainland Europe, is that it also enjoys being an island. And it's well known in ecology and in terrestrial biology that there is a concept out there called refugia. That is a refuge, right? You've heard of refuges like an animal refuge in the sub-Saharan Africa, for example. A refuge is a place where you get a lot of interbreeding and intrabreeding, so the gene pool tends to be condensed, so you'll get a lot of so-called outcrossing, right? But that can be both reinforcing and it can be detrimental to the species it's involved in that smaller gene pool sharing. And we'll get into a little bit about that. We're not going to get into any detail on this lecture because that involves individual genes and their structure. But we can talk about that in later lectures. At any rate, that island refugia might have something to do also with isolating that breeding population. That's what we mean by that. And in so isolating it, preventing maybe some parasites, pathogens, and whatnot that live and can move across borders in, for example, Europe, and spread out and cause famine and disease in the rest of Europe, but maybe can't cross that small bit of the Mediterranean so easily unless it's on shipboard. and therefore decreases the titer of maybe pathogenic bacteria or viruses, as well as maybe some parasites. So that's something to consider. Okay, the refugia, so it's an island. So let's go right into a little bit of genetics, because this is necessary for us to understand a little bit about the Sardinia and about this paper I'm going to talk about very briefly. What's a haplogroup? It's described in this paper, so I'm going to just dissect this and tear this down for you. First of all, we have to talk about a haplotype. A haplotype is a genomic cluster or cluster of genes in chromatin, where chromatin is made up of DNA and protein. And that haplotype is actually descended from a single parent. So I'm going to mention briefly and soon how we determine that. But first of all, let's use that definition to describe the haplogroup. Haplogroup is a collection of those haplotype clusters, those clusters of genes that can be traced back phylogenetically to a common ancestor. So now you have basically a cluster of genes. Okay, so it's the entire haplogroup. That's where we get the Neolithic farmer genome. That's what we mean by that. Okay. Then how do we go about examining that? Well, we get that by tracking single nucleotide polymorphisms, colloquially known as SNPs. Now, basically what those are individual locations in the genome, in the gene sequence, where there are alterations or changes in that nucleotide sequence. Remember, we have 4 nucleotide bases in DNA. So if you get a single change in the walking down that gene of a particular nucleotide, and that gene is something that gets inherited, and it's different from the backdrop of other people, we would call that a single nucleotide polymorphism. That is, there's a difference around that particular nucleotide. That's why we say morphism, or morph to a different nucleotide, is there? So if we study those in given sets of genes or clusters of genes, we can trace back to a common ancestor and say, aha, all the people that came from that ancestor have this number of SNPs in these particular genes. You see, that's how it's done, and it can actually be used to date. So SNPs represent a conserved, heritable single point mutation, or cluster of them in DNA, and they occur randomly. It's another important thing. At least as far as we can tell, there isn't any directed evolution here when SNPs arise. Now, there can, of course, be environmental aberrations, such as exposure to sunlight or maybe harsh organic chemicals that may enhance mutation rate, but where those mutations occur, that's the key there, in those particular nucleotides to generate that SNP, that seems to be pretty random. But anyways, once they're done, as long as they're not conferring some kind of morbidity or mortality to the cell that's inherited that SNP, and indeed that organism that keeps inheriting it from a common ancestor, then it's likely to be fixed in the genome. So the genome is pretty conservative. If you do get a mutation and the mutation doesn't cause any problems, to go back and change that mutation actually takes more biological energy and more sophisticated scrutinizing biological machinery to pluck out a change in a nucleotide, that the cell, or that is the genome, doesn't cooperate with that mutation in such a way so that it would remove it, because it actually could induce new mutations in the process of moving or removing a mutation that is benign or has no apparent effect. Okay, so that's why you find SNPs in the genome. So let's take a look at the Sardinia now and their haplogroup, which is what that paper was about, the paper I just talked about, 2017. The Sardinia specific haplogroup analysis from using the mitochondrial genome, a lot of reasons to use the mitochondrial versus the nuclear genome, and I'm not going to go into them here, but we can talk about that later. But you know you get your mitochondrial genome from your mother. You only get it from one parent. So it's more tractable in a way because of that, plus it's much less genes in the mitochondrial genome. So there's a lot of good reasons to use mitochondrial genome for these common descent studies, these phylogenetic studies. Again, I can go into that later. At any rate, looking at the mitochondrial genome of these Sardinia, we find 2 common ancestors, not one. We find Mesolithic, that's Middle Stone Age, and Neolithic, Late Stone Age, or New Stone Age, right? So that's not surprising, because indeed you see Mesolithic and Neolithic genomes in all humans that currently exist, but in varying percentages. That's the point. So what about the Sardinia? It seems like they have more of the Mesolithic than the Neolithic. Okay, so they have a higher percentage of the Middle Stone Age genome as recognized by SNPs in the mitochondrial genome than do, let's say, mainland Italians. And interestingly, mainland Italians have a little bit more Mesolithic than, say, mainland Germans or mainland Dutch. So it seems to be the closer you get to the Mediterranean and to the warmer climes, you get more of an influx of this Mesolithic genome. And that's probably because of migration of peoples using the seaway, right? Using the Mediterranean Sea, so you get more mixing of those genomes way back during the Mesolithic, which 10,000 years ago, roughly 10, 12,000 years ago is when agriculture was invented. And there was a big shift in genome when agriculture was invented from hunter-gatherer. So, as it says here, Neolithic varies with geographical regions, so you can't give it an exact date for Neolithic genome arising, but at least by 7,000 years ago, you had robust animal and plant husbandry, and that is then, of course, the invention of agriculture by humans, and by then we do have this on-the-scene new horizon of Neolithic genomic markers. Okay. So you have Mesolithic and genomic, and the Sardiniae seem to have more Mesolithic. So we can call that an unusual dual ancestral origin in Sardinia. So you have this fusion of Mesolithic hunter-gatherer genome and Neolithic farmers, and that created, of course, the European genome as we know it, but the percent, as I alluded to before, of the Mesolithic is higher in some regions. In Italy, it's higher than the rest of Europe, as I mentioned, and in Sardinia, it seems even more. So, can that relate to the longevity of modern Sardinians? This paper suggests maybe, and it goes into a lot of detail of specific gene clusters, specific haplotypes and haplogroups, which I'm not going to talk about today in this lecture. But again, if we dug through this, if we disambiguated that paper, if you came to us, to VerEvMed, with a question about aging and you wanted to know if you had, for example, an ancestry and you wanted to know how much Mesolithic ancestry you have, that is something tractable. And that may be useful to you to then look at the haplotypes and haplogroups in your particular genome if you had your genome sequenced. So we can get down to an individual level of discussion by launching from papers like this. And you don't have to be Sardinian to launch from it, of course, because it is a general principle of understanding of the mixing of the genomes, you see. So, can we say there are any general laws? Okay, so let's walk out of that paper a little bit and ask about general laws. There are vast differences in average and maximal lifespans, as we've been mentioning. And those differences span up to 5 orders of magnitudes. So that means they range from just a few days, where an entire lifespan will occur, to centuries. In fact, bacteria, they divide every 20 minutes, and so you could argue that their lifespan is 20 minutes. Okay? Although there are different sort of metaphysical principles you could argue against that. Anyways, the differences depend on developmental genetic programs. There's no doubt about that. Our genome is directed, right? It is directing a developmental program to make a mature organism in a given species. So you get direct growth, differentiation, and reproductive capacity within a species. And of course, this is well developed. This is what makes us a species, what makes humans different, for example, from the Neanderthal, at least as far as we understand the Neanderthal, or maybe even from previous common ancestors that go back to what gave rise to, say, the great apes or the monkey lineages. So what else can we say about this? This leaves the published literature lacking, okay, in general principles. So what we do is we take anatomical and physiological features of various species, and we say, does size matter? Does basal metabolic rate matter in a given species? Does that matter vis-a-vis to the aging process? And we can come up with these really big generalizations, and that's what you find in the published literature. For example, really large humans tend to die younger. Okay, so people that suffer from the generic term gigantism tend to only live maybe into their 60s, right? Whereas people that are of shorter stature may live longer, even within a given family. But there's certainly no direct causal link there, or even a direct one-to-one correlation. It's just a general observation, right? Really, really big doesn't necessarily mean living really, really long time. Maybe that's something to do with food resources and digestion, for example, metabolism, signaling through the bowel, signaling through the brain. All of that could be a component of that large size. Likewise, metabolic rate. Really high levels of metabolism tend to make people, I shouldn't say make, see, I made a mistake too, tend to be associated with people that live shorter lifespans. So a lot of cell division, a lot of protein synthesis within those cells, a lot of turnover, a lot of metabolism does not portend for a long lifespan, even in humans where it's been studied. So that's another general principle we can sort of take a look at. Now, is that a yardstick for understanding the physiology of senescence? Maybe not, but it's certainly something that we can use as a handle to pull out of these studies, to use as an intervening hypothesis, to ask questions, to devise experiments, to obtain results that turn into evidence that can be discussed, so that we get an idea whether or not the things we're looking at might be related to aging, right? So these are useful things to consider anyway. But at first glance, it looks like there aren't any really permeant biological laws. Permeant, by that I mean not permanent, but permeant, like all the way through all of biology. We say, oh, well, if you are larger than your brother or sister, you will die younger. It just doesn't, it's not that kind of tight correlation. And that's a very crude, non-robust physiological and anatomical parameter. But even if we look at, say, a given gene or cluster of genes, we can't come up with a completely permeant description and say, oh, well, you inherited this gene cluster and your grandmother died of complications of cardiovascular disease, so you will too at the age of 62, for example. They're just not those kind of correlations, right? It's just way too variable. There's too many variables even to be able to induce into one particular study. But we can talk about bioenergetics, that is energy, the production of energy in the cell. We can look at that, and I think that we will, throughout the course of these many lectures, examine whether or not that has a role. It seems to maybe have a role, as we'll see in a moment too. But what about defense too? Your immune system. If you have a strong immune system, you're going to ward off a lot of parasites and pathogens, and maybe even abiotic stresses. And those kinds of stresses, if you can survive them, will give you that survivorship principle, right? Will increase your chances to reach that maximum longevity. So we have to consider those things too. And the immune response is certainly not uniform, and it's certainly inherited, but it's also a memory-based learning biophysiological, biochemical, physiological sequence of metabolic pathways in terms of events. And so in real time, your immune system changes as you age as well, of course. So here's something interesting. Caloric restriction. Maybe you've heard this in the popular literature. Caloric restriction seems to postpone senescence in model organisms. This is something you see a lot in literature. So it's a suggested mechanism, but only suggested and only a mechanism, okay, to prolong life in lab animals where they give them severe caloric restriction. So when I say caloric restriction, I don't mean just withholding lipids, that is fats, or just withholding carbohydrates, or just withholding proteins, or altering the multiplicity of those three major food groups. We mean just restricting calories, all calories. When I say restricted, I mean cutting it in half, and then taking those animals and cutting it in half again. We're talking severe restriction, right? So rats reared on a low caloric diet, by this I mean cutting their calories in half throughout life, from when they're pups all the way until they're finally in old age, were, on the average, in a couple of studies that I've examined, 15% smaller. So there's that size differential. But interestingly, fantastically, they lived about 50% longer than their litter mates. And the litter mates were fed ad libitum, which means they ate whenever they wanted, whatever they wanted. Well, of course, it's probably mouse or rat chow for sure, because they're rats. But that just means that if you eat normally, and you compare that to severe caloric restriction, those animals that had severe caloric restriction lived 50% longer. That's pretty amazing. So when caloric restriction was imposed even later in life in these rat studies, okay, like in middle age for a rat, lifespan was extended, not that robust 50%, but up to about 20%. So that means if all of a sudden, this is a huge leap, right, extrapolation. If people decided to start restricting their calories at, say, the age of 40 or 50, it could mean an increase in overall lifespan, Okay, this would maybe suggest that. That's why you hear about fasting and about diets that restrict calories. That's where that kind of idea comes from. And it really is more of an idea, not quite built into a concept yet, because concepts need, I think, a lot more evidence than what we have. But we'll discover that together. So anyway, we can say that food-restricted rats also have less of these chronic diseases that tend to prevent you from reaching your maximum lifespan. What are those diseases? Well, cancer, cardiovascular disease, that is atherosclerosis in this particular realm of study, and also a lot of autoimmune diseases. And you hear more and more about autoimmune diseases, again, in the popular press and popular culture. Probably don't have much of a clue, unless you're an immunologist or a physiologist or biochemist, what that means, or if you're a med doc, surely you know about autoimmune diseases. But if you're outside the professional group that some of us reside in, you only hear about autoimmune disease. And I'm going to talk a little bit about that through these lectures, because it's a really fascinating new consideration when we think about biological aging, senescence. Okay. So, recent paper again, we looked at a paper in Molecular Evolution. Here's a paper in Cell, okay, published again very recently, just actually this month in March of 2017. That paper demonstrates that fasting restores pancreatic beta cells and thus is therapeutic for diabetes, both type 1 and type 2 diabetes. That's what that means, T1D and T2D, type 1, type 2. Now, type 1, remember, is an autoimmune disease, or maybe the first time you're hearing that, but it is. And type 2 diabetes is really looming large in the obese population and really becoming the predominant type of diabetes. And again, I have in mind giving a lecture on diabetes, and particularly about type 2, because it's such an important morbidity, mortality factor in human health. At any rate, this is fantastic news in a way. So seems like if you restrict calories, okay, fasting is restricting calories. Pancreas responds in these animals by increasing the number of beta cells. Why is that important? Beta cells, because this is an endocrine as well as an exocrine organ, that is the pancreas. The beta cells, of course, are the ones responsible for making insulin. So in both type 1 and type 2 diabetes, eventually you get insulin insufficiency. Okay, in type 1, there is an autoimmune destruction of the beta cells. You don't make enough insulin. That's why those people need to have insulin injections or patches. In type 2 diabetes, there's plenty of insulin being made initially, but you get insulin resistance. That is, the insulin that's being received by the peripheral cells, like the adipose and the muscle, does not get received by the receptor in those cell lineages. So the pancreas pumps out more and more and more insulin. Eventually you get like what's called a feedback inhibition, for lack of a better term, not to be too specific here yet. And because of that, you do get beta cell senescence. Okay, they stop producing as much insulin. And so indeed, in later stages of type 2 diabetes, insulin injections or patches are also recommended and prescribed by medical docs. So it's all basically centered around whether or not the pancreas is frankly biologically active and can rebound to the vagaries of what's going on with the system. Maybe it's environmental, like too many calories, for example, like this suggests. And what's amazing is that you have a replenishing factor there. You restrict calories, and the pancreas starts making more beta cells. And indeed, the payoff for that, right? The payoff, for that is not only do you make more beta cells, they make more insulin apparently, and not only do they make more insulin, they tend to help ameliorate both type 1 and type 2 diabetes in the system. So that's a really interesting paper that came up on my web feed just recently, and I wanted to make sure to share that with you. So here's another paper published just recently, in fact, just a few days ago, because we're in late March of 2017 when I'm recording this. And this is in the journal Science. And there's the citation if you want to take a look at that paper. I'm not sure if it's behind a paywall. Science tends to be, but sometimes they have open access for certain papers. So this paper suggests that a compound called NAD+, that's the specific chemical signature, enhances DNA repair by preventing an inhibitory protein from binding to a DNA repair enzyme complex. Now that seems like a mouthful if you're not a biochemist or if you're not a medical doc. Let me just explain it to you real briefly. NAD is a common metabolite. It's very robustly common in all cells in the body. It exists in two forms, the one shown there, that NAD+, which is known as the oxidized form, and NADH, which is the reduced form. The H is just a hydrogen atom, actually. And going through NAD to NADH, going through that change, and then NADH back to NAD, that's a redox reaction, right? Reduction oxidation. You're either stripping away electrons or you're adding electrons. When you add electrons, you reduce. When you strip away electrons, you're oxidizing. And that redox is how you make biological energy, in particular ATP. So NAD is around after you've used the reducing power of NADH to synthesize ATP through something like the Krebs cycle, right? The mitochondrial Krebs cycle that's hooked up to a series of enzymes called dehydrogenases, which remove that hydrogen, make that NAD+, right? So they oxidize it. And in the process, in that process, they reduce NAD to NADH because they're oxidizing keto acids. So in the TCA cycle, you introduce reduced carbon, and you oxidize that reduced carbon all the way to carbon dioxide. And in the process, you take NAD+ and make NADH. And that NADH then, again in the mitochondria, that's when it's reoxidized in something called oxidative phosphorylation. And it's named for what it does. It oxidizes NADH to NAD+, and it makes ATP, and ATP, again, is the energy currency of the cell. So you make NADH in the TCA cycle, and then you oxidize it in the oxidative phosphorylation pathway. So that's a redox couple, we call that. So there's a lot of NAD+ around after it gets reoxidized, and it gets reoxidized, right, by transferring those electrons basically to molecular oxygen to make water, and in the process it makes ATP. Okay. All right. Now that's interesting because what that means is that as NAD builds up, the DNA repair enzymes in the nucleus will go to work. DNA repair enzymes are a good thing because they will repair mutations that mount as the cell ages. So a healthy DNA repair system in your nuclei of your cells will enhance the lifespan of that cell because it will diminish the number of mutations that are allowed in that DNA. And DNA picks up mutations as it ages within the cell. So that's an interesting parameter there. Now, it was previously demonstrated that this same compound, NAD, that's nicotinamide adenine dinucleotide oxidized form, those of you know that, you know that's what it means, and those of you don't, now you do. There's also an enzyme called the deacetylase that uses NAD. That deacetylase has a cool name. It's called a sirtuin. There's multiple forms of those things. I mentioned them actually in a previous VerEvMed lecture. But anyways, what they do is they remove acetate. That's just a 2-carbon compound, again, common in all cells, very common. And when it removes acetate, covalently bound to histones, which are proteins in DNA, chromatin combinations, that is DNA protein combinations and chromatin in the nucleus. When you remove that acetate, it collapses that DNA, and when it collapses it prevents it from being transcribed, so you get less gene expression. So increasing NAD decreases the amount of gene expression. That has also been associated to improvements in longevity. Okay, so now you got a double shot of goodness from NAD, the oxidized form, right? So remember, you make the reduced form in the Krebs cycle, and then you reoxidize it in the inner membrane of the mitochondria during oxidative phosphorylation. So you're learning so much about biochemistry just in a few minutes. So that's a bioenergetics, basically. A lot of other things are going on in the TCA cycle for sure, but that's your bioenergetics. It's all in the mitochondria. Hey, didn't we just talk about the mitochondrial genome? Yeah. Some of those genes involved in this? You bet. Okay, so that's interesting too, maybe. I don't know. Anyway, condensed chromatin, which is what you get when you remove that acetate. You remove the acetate by having an NAD-associated deacetylase enzyme. That's going to give you a decrease as a good substrate for gene expression. And that looks like a positive thing for increasing lifespan in model organisms. There you go. So what can we conclude from this about NAD? Based on this paper recently published in Science and all the other evidence, some of which I just briefly alluded to, but which we could do in detail, of course, there's a limitation on gene expression that seems to trigger cellular fuel and therefore bioenergetic changes. Okay? And that maybe leads to less cell division, more cell maintenance, more DNA repair. And all of that would be proactive for increasing a healthy lifespan of a cell. So maybe less is more, less metabolism is more, less cell division, less bioenergetics, less overall energy, right? And certainly less gene expression, more DNA repair. Looks like that can enhance lifespan. So that's the end of the first lecture on aging. That is senescence. And we come from, again, VerEvMed. There's our website. There's my email address. You can contact me directly there and we can hook up and we can start discussing questions or problems you may have that you'd like for us to take a look at in depth in the published scientific literature. So again, what we are scientists verifying published evidence in biochemical, physiological, pharmacological, and of course the medical sciences. Thank you very much for your attention, and I hope you have a pleasant and productive day. Bye-bye.